Okay, let's see now the cell nucleus that contains the genetic material necessary to produce a whole human being. Have in mind the objectives when you review this lecture. Alright, you already know that the nucleus has a nuclear envelope that separates its content from the cytoplasm. The molecule that codifies the genetic information is the DNA, which stands for desoxyribonucleic acid. You also know from the first lecture that eukaryotic cells are the only ones to have a nucleus. The discovery of the structure and synthesis of DNA and RNA was a milestone in the history of medicine and opened a wide range of new diagnostic and treatment techniques. But who made such important discoveries? This is general culture. And how do they call the structure they discover? Okay, so how DNA and RNA were synthesized was discovered by the Spanish doctor Severo Ochoa and the North American Arthur Kornberg. They got the Medicine Nobel Prize in 1959 for their discoveries. Then Watson, Crick and Wilkins got the same prize in 1962 for the molecular structure of DNA that they call the double helix. Here you have Wilkins is less known, but he used X-rays to discover the structure of DNA. DNA is a very long string of nucleic acids. A single nucleus contains about 2 meters of DNA, which is packed by special proteins in the form of chromatin, and when they get very dense, they are called chromosomes. Humans have 23 pairs of chromosomes, 2 of them being sexual chromosomes, and they are named X and Y. When a cell divides, it has to copy all its chromosomes and then split them equally among the 2 daughter cells, as you see in the left image. When this happens at division is when the chromosomes are so densely packed that they get visible with a microscope, of course. If a cell is not dividing, we say it is an interface, and chromosomes are not so packed because the cell needs to read them. Alright, so at interface, the cell is doing whatever it's meant to do. Its function will differ whether it's a liver cell, an immune cell, or a gland cell. The proteins to do their duty are codified in DNA and synthesized in the cytoplasm by ribosomes. These ribosomes are made of RNA and produced in the nucleus. And after production, they tend to accumulate in a dense region of the nucleus that we call the nucleolus. Okay, so inside the nuclear envelope, we have the nucleoplasm and the chromatin. And it can be observed with a microscope because the chromatin especially dies very well. During interphase, chromatin that is being read tends to be near the nuclear envelope. This is because when special enzymes read and translate the DNA code, they produce a messenger RNA that has to leave the nucleus through a special passage that we call the nuclear pore complex. In order to read the DNA or chromatin, it needs to be accessible, so it must be extended. So we can distinguish 2 types of chromatin in a nucleus. Heterochromatin is the condensed version. It is easier to see as dense patches, as you see in the image. And since it can't be read, we say that is transcriptionally inactive and tends to accumulate in inactive zones that we call B zones. All right, so the opposite is euchromatin, which is transcriptionally active. In other words, this is being read for transcription or duplication before cell division. Not all DNA is copied at the same time. The nucleus has zones, we call zones A, where DNA moves to be transcribed. Here you have an image of a chromosome with its different sections, but don't worry now, we will come back to this when we get to the cell division lecture. And here you have a table displaying the main differences between heterochromatin and euchromatin. Basically, the biggest difference is active and non-active, okay? Let's go now with the nuclear envelope. First, let's say that this is a double membrane with a space inside. It is full of nuclear pores, as you see in the left image, and these pores are the ones that control the passage of big molecules like RNA. So it has a very important control role. The nuclear pore is a complex structure made of different proteins, as you can see in the right image. And we know that the space between the double membrane that makes up the nuclear envelope is about 200 angstroms and is full of new proteins. You can see in the right image marked as 1, the yellow 1, that's the double membrane of the nuclear envelope. And this is full of proteins because proteins are synthesized by ribosomes, that you see those small black dots all around the outer membrane. Those are ribosomes that are inserted in the rough endoplasmic reticulum, and these are the machines that read the messenger RNA and produce the proteins. So because this is connected with the nuclear envelope, as you see in the image, is full of proteins. Okay, here you have it marked, the rough endoplasmic reticular, as 2. Sorry. Okay, next one. The question is why the nucleus has that perfect round shape. I mean, why is round? It's round even in triangular or square cells. And the answer is because the nuclear envelope is stabilized by a net of intermediate filaments that we call the nuclear lamina. You can see it in the image as a dark gray line just under the nuclear envelope. Okay, let's go with the tough part of this lecture. As said, big molecules like proteins or RNA need to pass through the nuclear pore complex to go to the cytosol where proteins are produced. This pore is quite big but does not allow the free diffusion of molecules bigger than 9 nanometers. But the good news is that the pore can stretch and let particles up to 25 nanometers to go through, provided they carry a proper, let's say, passport. Let's see what is that. Here we have several images of a pore that is made up of 8 subunits making a ring. The ring, as you see in the lower image, projects inside the nucleus, looking a bit like a basketball basket that becomes narrower at the bottom. The final efficient diameter of that basket is only 10 nanometers. So molecules can diffuse through it more or less quickly depending in their size. But any molecular weight above 60,000 Daltons or above can't diffuse. So they need help for transportation. These big molecules, like proteins controlling the cell cycle or cell death, that we call it technically apoptosis, need to be transported. They need to be transported to the nucleus to start apoptosis. So this means that ultimately the pore complex is controlling all those processes. So it is very important. Let's see how this transport happens. Okay, so trade across the nuclear envelope is a complex process, but we will see here just the fundamentals involved, okay, because it's very complex. All right, so the nuclear pore complex has receptors that can interact with the load to be transported, and there are 9 different importins and 5 exportins. These are like passports to go in or to go out, but they are different. Loads may have a nuclear location signal as a label saying that they should be inside the nucleus. So if they are found in the cytoplasm and they have that signal, then they are transported inside. But they also may have nuclear exportation signals if they are inside the nucleus. That means that they must exit the nucleus. The big loads with importins or exportins can make the pore expand and let them through. And here is the difficult part. Okay, a protein called RUN-GTP is the one that controls the direction of transportation. It is found only inside the nuclear envelope. When a load binds to an NLS signal, it is imported into the nucleus, and once there, the RUN-GTP releases the load from the importin, and that importin returns to the cytosol. When a load binds to a NAES, that means it has to be exported. Then RUN-GTP attaches and exportation takes place of that complex. After exportation, once out, the load is released in the cytosol by RUN-GAP, and RUN-GDP results and detaches. All right, so GTP has been converted into GDP. The exportin returns to the nucleus, but RUN-GDP has to bind to NTF2 to go back to the nucleus. Once there, RUN-GEF converts the RUN-GDP into GTP again, and it can be used in a new exportation. All right, so since this is a complex process, here I leave you with a graphic animation to help you understand. Sorry, not this one.
Protein sorting between the nucleus and the cytosol is termed gated transport, with the gate being the nuclear pore complex, or NPC. The nucleus has a double membrane surrounding it called the nuclear envelope. The nucleus allows passive entry and exit of small molecules, smaller than 5,000 Daltons, but controls the passage of macromolecules like proteins, many of which are in the range of 25,000 Daltons, as well as DNA and RNA polymerases, many of which are around 100 to 200,000 Daltons. Ribosomes, which are huge, actually get assembled in the nucleus and must then be exported. Being 30 nanometers across, they can't get back in from the cytosol. The nuclear pore complex contains channel nucleoporins with large unstructured regions creating a messy tangle. This is why macromolecules can't freely come and go, but small molecules can. In order to enter the nucleus, a macromolecule needs a nuclear localization signal, or NLS, and in order to exit, it needs a nuclear export signal, or NES. With active transport, molecules up to 39 nanometers across can be carried through the nuclear pore complex. Protein cargo destined to be transported into the nucleus has a nuclear localization signal rich in positively charged lysine and arginine amino acids, which allows it to bind to a nuclear import receptor, also called an importin. Different cargo proteins can have different nuclear localization signals and can therefore bind different importins. An adapter is sometimes necessary to help the importin bind the cargo. The importin binds to nucleoporins at binding sites with many phenylalanine glycine repeats, termed FG repeats, in the nuclear pore complex and is transported into the nucleus. The nuclear export signal on macromolecules destined to leave the nucleus has a different amino acid sequence than the nuclear import sequence. The nuclear export receptor also binds to nucleoporins in the nuclear pore complex and is similar in structure to the nuclear import receptor. In fact, they are both encoded by a gene family called the karyopherins, or nuclear transport receptors. Now, let's take a look at how nuclear import works. A macromolecule has a nuclear localization signal and is hence destined to be transported into the nucleus. It binds to an importin, and the bound importin binds to nucleoporins of the nuclear pore complex. The cargo-bearing importin is shuttled across. At the other end of the nuclear pore complex, there is a RAN-GTP. RAN-GTP binds the importin, which has a sort of stacked alpha helix motif, forming a spring-like structure. The spring-like structure has a conformational change, and the cargo protein is released into the nuclear lumen, which is its destination. After this exchange, the importin bound to RAN-GTP is transported back into the cytoplasm. RAN-GTP is hydrolyzed to RAN-GDP and inorganic phosphate, and dissociates from the importin. The importin can now be reused to transfer the next cargo. For simplicity's sake, I omitted a few details in the previous explanation. There are a few more players involved in nuclear import and also export. Firstly, RAN is inefficient at GTP hydrolysis. Hence, RAN-binding protein, RBP, not shown here, and RAN-gap must work together to hydrolyze GTP and release the nuclear import receptor. An easy way to remember RAN-gap is to think of it as, literally, creating a gap where there used to be a phosphate group. Secondly, we have nuclear transport factor 2, or NTF2 for short, which transports RAN-GDP into the nucleus. Inside the nucleus, we have RAN-GEF, guanine exchange factor. RAN-GEF is bound to chromatin and catalyzes RAN to exchange GDP for GTP. Upon completion, NTF2 returns to the cytoplasm. Here's a brief overview of import. Feel free to pause the video and read. Nuclear export is very similar to import, so I'm going to cover it quickly. A nuclear export receptor binds to nucleoporins and is transported into the nuclear lumen. There, it binds with RAN-GTP, which then promotes binding with its nuclear export signal-bearing cargo. The receptor again binds to nucleoporins and the whole thing is transported to the cytosol. RAN-gap helps with hydrolysis of RAN-GTP to RAN-GDP, and RAN-binding protein helps detach RAN from the receptor. The cargo dissociates and the nuclear export receptor is free. Now the receptor returns to the nucleus without any enzymes helping it.
Okay, so that was all for today. The next lecture is going to be about the mitochondria and cellular respiration. So I see you then.